Okay, so you see that trend towards reduction of the gametophyte and dominance of the sporophyte as plants get larger and more complex. Remember that in seed plants, the gametophyte generation is not a free-living structure, but is rather a microscopic cluster of cells that is entirely enclosed within the tissues of the larger, more dominant sporophyte generation. When we get into angiosperms, you will see just how small that gametophyte generation can be. So seed plants produce megaspores and microspores. They're said to be heterosporous, meaning they produce two different types of spores. And the difference there is gender. The megaspores are female, the microspores are male. They will eventually develop into gametophytes that will produce eggs and sperm. Obviously, the female ones produce the eggs and the male ones produce the sperm. So when you have heterosporous plants, you see the production of these two different types of spores, which lead to the two different gametophyte structures.
Does one plant have both?
We'll get into that later. Usually yes, but not always. So the big thing that distinguishes seed plants from other plants are obviously seeds, right there in the name, and also pollen. Pollen is very important. The two of these things, what they really allow for is reduced dependence on water, primarily for reproduction and for development of the embryo. So in aquatic plants, you would see these flagellated sperm that will swim through their aquatic medium and find the eggs. And you still see that in a lot of the lower plants like the bryophytes, the mosses, and the liverworts. They still rely on a very wet environment for reproduction because the sperm still has to travel through water. In the case of these more derived plants that produce seeds and pollen, now they don't need a liquid medium in order to transfer their gametes. So who in here has seasonal allergies? No idea. Have yours been worse this year than usual? Yeah, mine definitely have. Because of all that rain we've been getting, a lot more stuff is blooming. Has anyone gone out to see any of the super blooms? Anyone braved the crowds at Lake Elsinore? I was not willing to deal with that.
I actually went to Lancaster, and you could go right off the freeway. You see them off the freeway.
That's cool.
Yeah, I went to Lake Elsinore as well, and that was like a 4-mile hike to get there. So,
well,
Lancaster's way better.
Interesting. I wonder if there's any of them left, because I wanted to see them, but I don't want to deal with the crowds.
I just went to get it.
Oh, cool. Well, maybe I'll get to speak with them. Okay, so where was I? Oh yeah, the reason you have these seasonal allergies is because these plants have evolved this method of breaking their dependence on water for transmission of their gametes. What do they do with them instead? Yeah, in many cases they're just released into the air. Most pollen grains in gymnosperms are shaped in such a way that they're really good at gliding through the air. A lot of them have wing structures on them. So they use the air to transmit their gametes rather than water, and they also end up in your eyes and in your nose and cause watery eyes and itching and sneezing. So the pollen grain, what that actually is something that encases and protects the male gamete. So it's encased in that sporopollenin that I mentioned, the really tough, difficult to break down substance that some plants can make, and that prevents desiccation. That prevents the gamete from drying out. Pollen can also travel great distances without water. In many cases, it's carried on the wind. In many other cases, it's carried by some sort of animal. The seed then encases and protects the embryo and also nourishes it. The seed will usually contain some sort of food for the developing embryo, which allows it to stay alive without germinating for a while if necessary. Sometimes seeds can survive for years or decades or even longer before germination. The big advantage here is that it allows the seed to just wait until conditions are more or less optimal for germination. It doesn't have to start growing unless it knows it has all the things around it that it needs to grow. This also allows for much longer dispersal of the embryo. Since the seed can survive without drying out for long periods of time, it can travel long distances. So this allows for much greater dispersion of offspring, I guess you call it, than you can find in the non-seed plants. So first, the gymnosperms. These are the first seed plants to evolve. Gymnosperm, if you translate it into English, it roughly means naked seed. This is a paraphyletic group. Do you guys know what that means? Remember? So what's monophyletic? Yeah, they've all descended from a single ancestor. In a paraphyletic group, that means they have not all evolved from a single recent common ancestor. So it seems likely that seeds probably developed independently in a few different lineages. There are about 1,000 described species of gymnosperms, so really not all that many when we consider that there are almost 300,000 total described species of plants. And they're broken up into 4 major lineages. The largest and most successful of the gymnosperms is the conifers. And you have the cycads, the ginkgophytes, and the neophytes, the silent G. So conifers, this is what most people would think of when we're talking about gymnosperms. They look like this, tend to be very large trees. They're the dominant phylum, and most of them are rather large trees with either scale-like or needle-like leaves. Now these leaves tend to be pretty well adapted to the environments they live in. For instance, in the environments where you find a lot of these conifers, you also tend to get snow in the winters. The really narrow needle-like leaves are an advantageous adaptation there because they don't trap as much snow on top of them, thereby reducing the weight the leaves or the branches have to bear in the winter and reducing breakage. They're also very dominant in high altitudes and cold climates. One of the really cool things that needles are also good at is filtering fog out of the air. That's why you tend to see these in temperate coastal environments. It's actually really kind of cool to watch where the fog comes in from over the ocean and hits this big forest of coniferous trees, and the needles will filter those fine droplets of water out of the air, and it just starts raining down on the base of the tree. It's cool stuff. Now, most of these are evergreens. You guys all know what evergreens are? Can anyone give me a concise definition of an evergreen?
Always green.
They don't drop their leaves at any point during the year, so they always have their leaves. The opposite of evergreen would be deciduous. Those are trees that do drop their leaves in the winter. So evergreens, things like pines and spruces and firs, redwoods, sequoias, yews. So conifers are the largest trees in the world. Remember I mentioned that we have both the largest and the tallest tree in the world here in California. The largest by volume being a sequoia, the tallest being a redwood. I think that redwood is something like 350 feet tall. That's an incredibly tall tree.
Do we know if the conifers came first as like the
I don't actually, you mean were they the first gymnosperms to evolve?
Mm-hmm.
I don't know that.
because they're pretty big.
My instinct would be to say no for that exact reason, but I don't know that for a fact. There are a few, I mentioned that most of them are evergreen. There are a few examples of deciduous conifers. Those include the larch and the tamarack. Those tend to be found in colder, higher altitude environments. So there are a couple of conifers that do drop their leaves in the winter. Another group of conifers is the, or not conifers, of gymnosperms is the cycads. These tend to be much more successful in mild climates. Now, what does that look like to you guys?
It's like a pine.
It looks like a palm tree with pine cone, which is basically what cycads are. So they're often mistaken for palms because they have very similar leaves. They have those large, palmate, compound leaves that you see on palm trees. But they also have that gigantic seed cone in the middle, which separates them from the palms. That's how you can tell the difference. Now, these were dominant during the Mesozoic era, about 251 to 65 million years ago. But they've really declined since then, and they're actually facing the possibility of extinction now. what's sort of saving this lineage of gymnosperms is that a lot of people plant them as decorative plants in their yard. Moving on to the ginkgophytes. If that sounds familiar, you've probably heard of the only existing species of ginkgophyte. Yep. It's the only species left in this entire group. It's also the only seed plant that has dichotomous venation in leaves. Another thing that's interesting about the ginkgos is they have entirely separate male and female trees. So even though most seed plants will have both male and female organs on the same plant, not all of them do. People plant these as decorative plants because the leaves are kind of pretty. They have a really nice soft green color. But people usually only plant these.
Sorry, so in the ginkgophytes or whatever, there's a gene that codes for sex in that plant?
Well, there's a gene that codes for sex in all other plants as well. It's just the different reproductive structures tend to be found on the same plant. So if you look at any seed plant, the gametophyte structures will be either male or female, not both. But they tend to be housed on the same individual. In this case, it's sort of a relatively unique thing where they're only found on specific trees. You have a male tree and a female tree. And people tend to only plant the male trees because the female tree produces a fleshy seed that smells like rancid milk. Yeah. Yeah, if you pick one up and like squeeze it in your fingers, you will have to wash your hands all day to get rid of that smell. The interesting thing about that is the fleshy seed is not a true fruit, even though it looks like one. So there's a lot of weird stuff about the ginkgophytes.
Is ginkgo biloba the only example that we've been able to study on the ginkgophytes?
It's the only example we've been able to study alive. We may have other ones from the fossil record. Then the kmetophytes. These are the closest within the gymnosperms to the modern angiosperms. They have broad leaves. And most of these exist as vines in tropical and subtropical zones. But there are desert-growing species. In fact, there's a desert-growing species found in the deserts of Namibia and Angola that can live for up to 2,000 years. Tiny little bush, around for a long time. This image on the right here comes from the genus Ephedra, also called Mormon tea, which grows in dry areas of the southwest US and Mexico. What do you think we make from these guys? Based on the genus. Ever heard of a drug that sounds like that? Ephedrine? Pseudoephedrine is a synthesized version of ephedrine. You can make drugs from those guys. Yes?
They figure out like the last 2,000 years.
My guess would be that they aged living specimens. So that's how we know the age. With plants, it's easier to tell the age of things because they'll usually deposit rings in their structures. Have you guys ever seen that happen where someone, cuts a horizontal section out of a tree and counts the rings? You guys know why those rings form?
Because of weather, right?
Yeah, it has to do with changing growth conditions throughout the year. So when conditions are really favorable to growth, usually in the spring and summer, it deposits less dense woody tissue in that ring. When the conditions get less favorable in the winter, it deposits more dense woody tissue in that ring because it's growing slower and it tends to look darker. So that's why trees form rings, and that's why we can usually estimate how old these plants are. Now in the case of this bush, it might be very difficult to count those rings, but I would still assume that's how it was done.
You would just need to cut a part of the face, right?
Probably. And you probably also have to use a microscope. You can do the same thing with aging fish. If you take the bone out of their ear, they also deposit rings in there, and you really need a microscope for that. You have to cut it with a diamond saw and polish it and look at it under this microscope and painstakingly count these rings. It's really annoying. Ichthyology is great. Anyway, so this is the gymnosperms. Any questions on these before we move on to their more derived cousins, angiosperms? Okay then. So angiosperms, these are the flowering plants. Now, we've done a lot of talking about plants, and we've seen a lot of fairly broad diversity so far, looking at the bryophytes, and seedless vasculars, and the gymnosperms. But we've still only accounted for about 20 to 30,000 species of plants so far out of the 300,000 we described. So that would lead someone to the conclusion that the angiosperms are by far the most diverse lineage of plants, and probably also the most successful. Because how would you measure success in biological terms? Probably diversity and sheer numbers. So there's around 260,000 species of angiosperms. That's nearly 90% of all plant species that we describe. And they dominate most terrestrial ecosystems. When you look out at almost any patch of nature, if there's lots of plants on it, it's likely that most of them are going to be angiosperms. Now they have two structures that are unique to this group, which are responsible for their incredible success. Who can tell me what those are?
Well,
Yeah, first one's pretty obvious, flowers. What's the other one? What else do angiosperms produce that no other lineage of plant does? Fruits, very good. So flowers and fruits are only found in angiosperms, and these two adaptations are largely responsible for their incredible success. Largely through the formation of symbiotic relationships with animals. So remember that reproduction was one of the main challenges faced by plants as they transitioned from the aquatic to the terrestrial environment. So these guys, the angiosperms, have come up with a really clever solution to that problem by using other organisms to help them reproduce more successfully.
so instead of deterring predators, they're more promoting predators.
Well, they do both. So they're deterring herbivory of their general tissues in many cases, but in many other cases trying to encourage the consumption of their fruits. So it's sort of like, hey, you know, don't eat my body, but here you can have this, you know, big pack of sugar. So flowers themselves are incredibly diverse. They come in all shapes and sizes. This is just a cool old drawing of some domesticated flowers in England. But even in this incredible diversity of flowers, they all serve the same purpose. That purpose is reproduction. So the entire point of these flowers, the whole reason why these plants put all this energy into developing these really beautiful structures, is to aid in reproduction. And even though they look incredibly different, like you can think of something like a tiny little dandelion that we consider an ugly little weed, or you know, beautiful roses or gigantic magnolia flowers that are almost the size of a volleyball, they all share the same basic structures. Those structures being sepals, petals, pistils, and stamens. I'm going to go into a little bit more about what each of these are. So sepals, these are tiny little leaves that exist at the base of the peduncle or the stem of the flower. They exist in a single whorl, meaning they all come out of the stem at the same point. And that whorl of sepals is something that we call a calyx. So there's a developing rosebud with the calyx that has recently opened. Before the flower blossoms, the calyx entirely encloses and protects the bud. Now these sepals are usually photosynthetic. There are a few exceptions to that though. I think tulips have non-photosynthetic sepals. In fact, I think in tulips, the sepals are almost indistinguishable from the petals. Now the petals themselves, they exist as a whorl inside the calyx. So they also originate from the same point in the stem. This whorl of petals is called the corona. So in this diagram here, after the bud has opened, you can still see the sepals existing here at the base of the flower, Then inside this whorl of sepals, you have this other whorl of petals. Corolla is supposed to be corona. Or maybe I'm wrong. Double check your textbook on that one. I got this diagram from a different source. Anyway, the petals are usually very brightly colored. They often have very vivid patterns of color. What is the purpose of that?
Attracting animals like these.
Exactly, that is to attract pollinators. In most cases, the flower is primarily designed, or at least the petals of the flower are primarily designed to attract pollinators. Pollinators are often species specific, which makes this a very advantageous adaptation. Because you have one single species of animal, usually some type of insect, possibly some type of bird or rodent or bat, that will specifically visit your species of flower because it likes the petals you make and it likes the nectar you produce. When it comes to get that nectar, you're going to dust it with your pollen, and you can be pretty confident that the next flower it goes to is also going to be a member of your species. So it's relatively targeted delivery of pollen to other flowers of your own species. Very efficient.
Are bees generalized pollinators?
Some bees are, but a lot of bees will more specifically visit certain types of flowers than others. Same thing with other insects. Butterflies and moths will generally have a broad type of flower that they like to visit. It's usually based on color and scent. Now there are some cases where flowers are not pollinated by animals, and in that case, the flowers generally tend to be very small and very dull. Because if you're just going to use the wind anyway, what's the point of putting all this energy to make it look pretty if no one's coming to visit you? Now the sexual organs, which are potentially the most important part of the flower, are located at the center of the flower. So we've looked at the calyx and we've looked at the corolla or corona, still not quite certain on that, which is the petals. In the center you have the sexual organs. In the very center, you generally have the female organ, which is called the carpel or pistil, or sometimes also called the gynecium. And this contains three main parts. It has the stigma, the style, and the ovary. There's that larger word that sometimes is used to describe as well. So is this sporophyte or gametophyte tissue? Yeah, it's still sporophyte tissue. Remember I mentioned that the gametophyte gets drastically reduced in angiosperms, and you're about to see how drastically. So the megaspores that are produced in this tissue would then lead to the gametophytes. They're produced in the thick tissues of the carpel and protected by it. If we look at this diagram here, here's our cross section of a flower. This part right here in the middle that looks kind of like a big vase, that is the female structure, the carpel, or also called pistil, or also called gynecium. Contains the stigma, style, and ovary. The stigma is near the tip. The style is this long tube in the middle, and then the ovary is this bulbous part at the bottom, which contains the megasporangium. The stigma is the part where the pollen enters, and it's usually sticky to help facilitate that. When a bee carrying some pollen on its leg comes and brushes up against you, you want to grab as many pollen as possible to have the highest likelihood of ensuring fertilization. So that's why the tip of that tends to be rather sticky. The style then connects the stigma and the ovary. So you have that sticky entering at the tip. The style forms a long tube through which that sticky opening and the important ovaries are connected. The ovary houses the ovules. Each ovule, after it's fertilized by pollen, will develop into a seed. So it's not a seed until after it's fertilized. Until then, it's just an egg. Any questions so far on the female parts of the flower?
So the female part, none of it is gametophyte at all?
Well, we haven't gotten to the gametophyte part yet. I'll get into more detail in this in a little bit when we talk about the life cycles. So then the male part of the flower is called the stamen. That contains the filament and the anther, so it only has two parts. Sometimes the stamen is also called the andrecium. Usually flowers will have multiple stamens or multiple andrecium. They will usually only have one pistil, but that'll be surrounded by multiple stamens. The microspores, which are the male spores, those are produced in the anther of the stamen, and that will develop into the pollen grains. So here we've got our big vase-shaped female part in the middle, and it's going to be surrounded by several of these male pistils. The pistil has the anther at the end, which is the sac-like structure, which is supported by that long filament. So inside the anther, that's where you have the microsporangia, which are producing the microspores, which are going to be gametified tissue, which will then later give rise to pollen grains. So the gametified structures in these flowering plants are incredibly tiny relative to the volume of the plant. And we go on to fruits. So the fertilized ovule inside the ovary will develop into a seed. As that seed develops, the ovary will enlarge itself. So the tissue surrounding that seed will also grow and develop as the seed itself grows and develops. Eventually ends up developing into a ripe fruit. This here is an example of the development of an apple. So here we have a flower that's been basically just been fertilized, and it's been cut in half here, and you can see the two newly fertilized seeds inside this ovary. Over time, as those seeds grow, the plant will deposit more and more sugars into the ovary tissue as well, causing it to enlarge, eventually developing into this large, delicious, sweet fruit surrounding these seeds in the middle.
Did that really grow, cut in half?
I'm assuming they just took a bunch of different ones at different developments of, or different stages of development. If they, I would imagine that the ones they cut in half probably died shortly thereafter. But what you see there is just the different stages of development. It's like when you go to a museum and you see the exhibit on human fetal development, and they have fetuses from each stage. Sometimes it's really creepy. I was in a museum in London, and they had a gigantic model of a human fetus. It was like bigger than me. Like, it was really awful. Anyway, there we have that giant delicious fruit. So it serves to not only protect the seed while it's developing, but once the seed is fully developed, it then serves to attract some animal who's going to eat that delicious fruit and swallow the seeds, and then later on place those seeds somewhere else with a neatly convenient pile of fertilizer. Now, as for the fruits themselves, they can be either fleshy or dry. Most of the things people think of when they hear the word fruit are fleshy fruits. These include things like that delicious apple or grapes or tomatoes, something like that. So fleshy fruits have this large, sweet, moist, fleshy covering around the seeds. Dry fruits, these are things that people don't tend to think of as fruits but are. Includes things like peas, includes okra, includes some nuts. Surprisingly, other nuts are actually fleshy fruits. Have you guys ever seen a raw almond, or like anyone gone picking walnuts or cashews? They develop these huge, large, fleshy fruits, and all we eat is the seed in the middle. Dry fruits also include many things that aren't eaten by humans, like this milkweed fruit that would probably be really disgusting if you tried to eat it. But it is a fruit nonetheless. So, looking at this, clearly not all fruits are designed to be eaten. Some fruits serve to disperse seeds through other purposes.
What would you like of that picture? What would that purpose?
We're about to get into that right here. So, the general purpose served by all seeds is to, served by all fruits is to help disperse seeds. there are several different strategies for doing that though. Some fruits are just carried by the wind. Anybody know an example of those?
Dandelion.
Absolutely. Dandelion fruits, those are fruits that are obviously not designed to be eaten. Those are designed to be carried by the wind and dispersed that way. Other fruits may be dispersed by water. Who can guess one of those? What? Perfect. Coconuts. So coconuts can travel from island to island because they have this large floating fruit that can carry it all the way across vast oceans. The more common method is that fruits are designed to be eaten by animals, which will then deposit the seed in some other place. Still other fruits are designed to hook onto animals' fur using burrs or hooks and be carried around that way. The sweet gum fruit is an example of that. So here you see a lot of different methods of dispersal, all served by different designs in fruit, but the main point is always greater dispersal of seeds. Does this make sense? When you have 260,000 different species, it's likely that they're going to develop a lot of different strategies for survival and reproduction. Yes?
You know what goat horns are? Little like things that speak.
Goat horns?
Yeah, they're like little like ball that have these like ones that.
Do they grow around here?
Yeah.
I've probably
Probably like
been stuck by them and just not know what it was called.
Yeah.
Yeah, they're like super annoying. They can get all over your clothes. and
Oh,
stuff.
yeah, definitely experienced those things. I just didn't know the name for them. Yes, those are fruits. All right, any questions on fruits before we move on to the angiosperm life cycle? We get to talk about immunophyte tissues more specifically. Okay, so obviously the sporophyte phase is dominant in these plants. You never really directly observe the gametophyte when looking at these plants. But like the gymnosperms, these are heterosporous. You guys remember what that means?
Yeah, both male and female.
Yeah, they produce two different types of spores, both male and female spores, microspores and megaspores respectively. I can't say megaspore without thinking of the mega fruit from Rick and Morty. I'm assuming most of you guys know what I'm talking about. Anyway, yeah, heterosporous means it produces two different types of spores. Maybe only a couple of you guys know. If you don't watch Rick and Morty, I probably shouldn't recommend that you do, but if you want to. Anyway, so the microspores, these are the male spores. They will eventually develop into pollen grains, and that came up out of order. That was supposed to come up first. So remember that all the male tissues exist in the anthers of the flower. You have microsporocytes that are in those anthers, which is still sporophyte tissue. The microsporocytes will divide by meiosis in order to produce those haploid microspores, which are finally now gametophyte tissue. The microspores will eventually divide and develop into pollen grains, and there you have your gametophyte. So each pollen grain here contains two cells. It has one cell called a generative cell. That cell will divide, resulting in two sperm cells. It has a second cell that will end up becoming the pollen tube cell. So if we look at it in sequence in our diagram here we've got our male part of the flower. We're going to focus in on that anther. And within the anther, we're focusing on those microsporangia. Those will produce microspores, which will then divide through meiosis, because we have to go down from diploid into haploid tissue here, resulting in a pollen grain, which will divide by mitosis to produce our generative cell and our tube cell. The generative cell then divides by mitosis, producing two separate sperm cells, and then we have our pollen tube cell as well. Now you may wonder, why 2 sperm cells in each pollen grain? Has anyone read far enough in the chapter to know that? Well, then I'm going to leave you on a cliffhanger and explain it a little bit later. But it is important. It's not just for overkill or to ensure greater odds of fertilization. That second sperm cell plays a very important role in angiosperm fertilization. Now the female stuff. All this happens in the female part, which is that carpel in the middle of the flower. Here you have megasporocytes, which are held within the diploid sporophyte tissue, which will divide by meiosis to produce exactly four haploid megaspores. This takes place inside the ovule. And of these 4 haploid megaspores that are produced, one of them will be larger than the other 3. That big one will end up dividing by mitosis to form the female gametophyte, which is also called the embryo sac. Three more mitotic divisions will end up producing 8 nuclei in 7 cells. This is also not a mistake. So you have 7 cells there. One of them is the egg, and that egg, along with 2 other cells, will move to one end of the embryo sac, while the other 3 cells move to the other end. 2 of those nuclei will remain in a single cell, so you have a dinucleate cell. They will then form to fuse a diploid nucleus. Remember, they were haploid before that. That diploid nucleus moves to the center of the embryo sac. We'll go over this part of the diagram before we move on. So here, in our female part of the plant, we have our ovary. Within the ovary is a macrospore, which will divide, forming these 4 megaspores. Here you now have what we see at the end of this process, where we have our egg and 2 other cells on one end, 3 cells on the other end, and in the middle, that big dinucleate cell, which will fuse to form that diploid nucleus in the middle of the embryo sac. That's important too.
So before meiosis, the 4 megaspores will, those are diploid and then they split into 8 haploid?
So those are diploid, but only one of them will split into those other haploid cells. So you have 3 divisions, 1 goes to 2, 2 goes to 4, 4 goes to 8 of that one cell. When a pollen grain then reaches that sticky stigma at the tip of the carpel or pistil, it will grow a pollen tube that goes all the way down the style and enters the ovule. It will then deposit both of its sperm cells in the embryo sac. Remember, it has 2 sperm cells, and you're about to find out why. Lucky you. One sperm will fertilize the egg, and because it's haploid joining with the haploid egg, we get a diploid zygote. The other sperm will then fuse with that diploid nucleus in the middle of the embryo sac, which forms a triploid or 3-end cell, which will develop into something called the endosperm. The endosperm is what later serves as that food source for the developing embryo. This is called double fertilization, and this is unique to angiosperms. You don't see this in any other lineage of plants. So you have that endosperm, which develops into a food source, which exists outside of the zygote. Go back here. So at this point, we have our diploid embryo here, and then outside of that is our triploid endosperm, and all of that is contained within the seed coat.
So what happens to the other cells that are not the egg or the diploid cell in the middle?
You know, I'm actually not sure of the answer to that at the moment. I would imagine, I probably shouldn't imagine, but that won't be on the test. Anyway, so the zygote then develops into the embryo, which has something called a radicle, which is a small root. It also has either one or two leaf-like organs, which we call cotyledons. This ends up becoming important when we try to further classify angiosperms. So remember that term. At this point, what the cotyledons do is they help to break down and transmit that food source from outside the embryo inside it so that it can use that energy. So the cotyledons are incredibly important in keeping the embryo alive while that seed remains more or less dormant. Okay, any questions on the life cycle so far?
We can't see the
In this image now, they're not. But just picture one or two tiny leaf-like looking things. So most flowers, in general, will have both the male and the female sex organs on the same flower. Those flowers are referred to as perfect flowers. Some plants, however, will have separate male and female flowers. Those are referred to as imperfect flowers. Now, in the case where you have separate male and female imperfect flowers, they may be found on the same plant, in which case that plant would be described as monoecious, or they may be found on different plants, in which case the plant would be called dioecious, meaning either one house or two houses.
So with perfect flowers, if you were to cover them, would they self-fertilize?
In many cases, yes. Now, a lot of plants have specific mechanisms in place to prevent self-fertilization, because that is like the ultimate incest, and it can often result in accumulation of undesirable traits in the offspring. So most plants will have some sort of mechanism in place to prevent self-fertilization. Some of them do use it, but most of them attempt not to. Now, in the case of perfect flowers, obviously the plant must be monoecious. You can't have male and female plants if the flowers themselves are both. In the case of imperfect flowers, where you have male and female flowers, then you can have either a monoecious or a dioecious plant. Does that make sense? Okay, great. All right, any questions on the life cycle? We've only got a few more slides, and we should have at least 20 minutes or so left for review. All right, so on to diversity. The angiosperms are classified as a single division, which is termed the Anthophyta. This is thought to be monophyletic, meaning it's thought that all flowering plants descended from the same ancestor. It's thought that this trait only arose once in evolutionary history, which is kind of crazy to think about. Also crazy to think about is geneticists think the same thing about blue eyes. Every person with blue eyes is thought to have descended from a single person who lived in Europe about 6,000 years ago. They looked at the math and decided that that mutation was unlikely enough that it probably only happened once, which is pretty wild. Anyway, so within the Anthophyta, we can divide it further into two major groups, the monocots and the eudicots. There's also a third little group, which we call the basal angiosperms, which we think branched off before the division between the monocots and the eudicots. We think this because basal angiosperms share characteristics with both groups. So these guys have things in common with both of the major divisions. They're also often referred to as the Magnoliidae because they include the magnolia tree. That's the most representative member of the basal angiosperms. Also includes laurel. It also includes things that aren't trees, like cinnamon or spice bush. And it also includes avocados. So basal angiosperms are great because they include avocados, which are awesome. If you don't like avocado, you are weird. Although I wish more people didn't like avocados, so it would be so expensive. Anyway, so moving past the basal angiosperms, the monocots. Can anyone guess where this name comes from? Who's been paying close attention to detail? What do you suppose is the defining characteristic of the monocots?
They only have one
They only have one what?
cotyledon.
Yes, one cotyledon. So monocots have a single cotyledon in the developing seedling. That's short for monocotyledon. Now monocots tend to be, within the angiosperms, tend to be what we would consider less derived or less complex plants. They rarely contain true woody tissue, only a few members. The vascular tissue in their stems is also generally not arranged into any particular fashion. The root system is also not arranged in any particular fashion and contains no major taproot. You guys know what a taproot is? So we see this in the eudicots, where the root system has one large dominant root in the middle that goes down really deep. We call that a taproot.
It's just has a bunch of little roots.
Yeah, it still has other roots as well, but it'll have one major central root, sort of like, and everything else kind of branches off from that. Now that is absent in the monocots. Now this lineage includes things like lilies, grasses, orchids, palms. The eudicots, in contrast, where do you suppose that name comes from? Not just multiple, be more precise. Two, yes. Remember I mentioned that developing angiosperms will have either one or two cotyledons, never more or less than that. So the eudicots have two cotyledons. Used to just be called the dicots, and I think that tended to include a lot of the basal angiosperms, but they then changed it to be the eudicot, meaning the true dicots. So, two cotyledons. They also have their vascular tissue arranged into a ring in the stem. So these tend to be more organized plants. They also tend to be able to produce woody tissues. When I say woody tissues, I mean cell walls that are reinforced with lignin on top of the cellulose. They also have a much more arranged root system, which is anchored with one large main taproot, which develops from the radicle. You guys remember what the radicle is? That tiny little root thing you see in the developing embryo inside the seed. So in the eudicots, that ends up developing into one large central taproot. Now the eudicots include 2/3 of all flowering plants. So it would probably not be inaccurate to say that this is a more derived or more advanced form, because it's more complex and it's also more successful. Okay, does anyone have any questions on any of this stuff?
Can you just go over cotyledons again?
Of course I can. So the cotyledons, when you have that, I'm gonna look at the picture that would make it easier. The cotyledons are in the developing embryo, and they really do basically look like tiny leaves. So here is a appears to be a lima bean before germination. Here you have the embryo, and here you have your cotyledons. Let me see if I can find something better than that. Ah, there we go. This one's much better. Yeah, it's a minute bigger.
Press Control Plus.
Control plus. Nice. I did not know about that shortcut. Okay, so here you have, is this a monocot or a dicot embryo? Dicot, how can you tell?
Two copies.
Yeah, we have our two cotyledons here. Here we also have the developing radicle, little root tip there. And here's the main body of the embryo with some shoot apical meristem right there that's not particularly obvious because it hasn't really grown much yet. So does this sort of clear up what cotyledons are? The main purpose at this point is to help the developing embryo access the food in this endosperm tissue, which is what takes up most of the rest of the volume inside the seed.
Is the root tip typically on the opposite side of the cotyledon?
Yes. Because when it germinates, the cotyledons are going to grow upward as leaves, and the root is going to grow down as root.
That's why that experiment that you talked about with putting seeds against the wall. And
Yeah.
having
You know, I'll see if I
different-
can find a picture of that too. I bet someone posted something like that. Geotropism experiment. Yeah, here we go. This is a lot like what it looked like when we did it. So you pin the seeds to some surface in different orientations. We have one right side up, one upside down, and one sideways. And no matter what, you always see the root come out and grow straight down. I couldn't explain on the biochemical level how the root senses what is down, but it has to do with gravity, and it does.
The embryo, is it floating in the seed? Like, can it move in the seed, or is it like in a
I would
fixed
assume it's
position?
relatively fixed in the same place, because I don't think the endosperm is liquid, so it wouldn't make sense for the seed to be, or the embryo to be floating around inside. Okay, any other questions? Are you comfortable with what cotyledons are now? Great. So we've got about 28, 27 minutes, it looks like, for review for our upcoming exam on chapters 11, 12, 13, and 14. Does anyone have anything they would like to go over?
The corona and the corolla, I found pictures on the internet that show both.
Okay, so in that case, I'll accept both on the test if there's a question about it. It's really annoying when that happens. Yes. What? Yeah, I guess we could do that. I don't have the whole thing written yet. Okay. So, let's go to the chapter 11 study guide. Alright. Number 1. That's probably too small for you guys to read. Let me just download the file.
You can always do Control Plus and zoom in.
Yeah, but I like this better. So, can you read that?
Yes.
And I'll just click on the plus there. Okay. So, number 1, who was Charles Darwin? That is not a good enough description of who Charles Darwin was. So Charles Darwin was a naturalist in the 19th century who is largely credited with being the first person to develop the theory of natural selection. How did he do that? Where did he go?
The main place to go.
Yeah, the the big place that really sort of sparked this theory was the Galapagos Islands, in particular those finches.
But he travels.
Oh, you went all over the whole world. In fact, that stop on the Galapagos was just one stop on like a 5-year cruise around the entire planet. And it was the only time he ever went there. And I think he was about 26 at the time. Alright, so people are comfortable with who Charles Darwin was and why he's so famous. How about number 2? What is artificial selection?
And selection.
Yeah, but how do we do it? How do we do this artificial selection?
We choose desired traits and we breed accordingly.
There we go. Selective breeding designed to enhance or diminish specific traits. So how does this then give us insight into natural selection? This is on campus already. So remember that On the Origin of Species, Charles Darwin spent a lot of the first part talking about artificial selection. This was to sort of demonstrate the concept of how specific traits can be either enhanced or diminished through relative success of reproduction. So it was a really brilliant way to frame that, because no one in their right mind was going to say, no, we can't enhance traits through selective breeding. That's impossible, because we've been doing it throughout all of human history since the agricultural revolution. So it was a great way to sort of introduce the concept of how genes can be either selected for or against and become either more or less prominent in a population. So this was the way that artificial selection really gives us insight into how natural selection can happen. So it sort of became almost, well, I don't know if obvious is the right word, but it really helped to sort of lay the groundwork, because in a matter of just a couple of centuries, we can see enormous traits, or enormous changes in traits through artificial selection. So if we're thinking on the scale of geologic time, then it becomes a lot more conceptually accessible how these large traits can happen through natural selection. All right. Speaking of natural selection, number 3, what is it? How does it happen? Why is it different from artificial selection?
It's the
environmental pressure.
Yeah, it's natural environmental pressures, which are either increasing or decreasing the reproductive success of certain individuals in a population due to the genes or the traits they possess. Now there's a couple ways it differs from artificial selection, one of which being that it's natural pressure rather than humans selectively breeding things. Another way it differs is that it's not goal-oriented. So whenever we selectively breed something, we have a specific end goal in mind. That is not the case with natural selection. Okay, number 4. How are you going to define evolution in biological terms?
the change in allele frequency in a population over time.
Perfect. That is a perfect answer. So it's a change in allele frequency in a population over time. Now, on the quiz, when I asked that question, a lot of people, I shouldn't say a lot, but several people seemed confused as to how you actually define a term, because they defined evolution as when organisms evolve. You cannot use a term in its own definition. That is not acceptable. So if I ask you to define evolution or any other term, never use that term or any of its root words in the definition. Imagine if the dictionaries did that.
They do. Have you ever seen the examples?
I have not. That was really
There's a bunch
frustrating.
of examples of like.
Well, I guess I've seen like if you were to like search like what does it mean when like define idiosyncratic, it would be like, you know, the state of being in idiosyncrasy. It's like, ah, come on. So then you have to go look up idiosyncrasy. It's not useful. Don't do that. Remember what he said, the definition of evolution. And if you can't remember it, it's right there in the PowerPoint slides from that lecture. Okay, number 5, understand the observations Darwin made that led him to his inferences regarding evolution and be able to explain what they mean. So what was that first observation?
Yeah, you're referring to the plants or the birds?
So I'm referring to life in general.
Okay.
So the first observation being that all species of organism are capable of producing more offspring than their environment can support. The second one being that different traits that are heritable will make things either more or less successful in reproduction. So understand what those are and what they mean. There was a true-false question on the quiz about whether or not all species can produce more offspring than the environment can support. That is absolutely true. And you see that in basically any population of any organism. Animal, plant, fungal, bacterial, whatever. Okay, number 6. The 3 key points that I emphasized about natural selection in lecture. So those are the 3 things that must be true in order for natural selection to act on trait. So the key points, let me just pull up that lecture so it'll be probably easier than listing it off. So the first one being that natural selection occurs through interactions between individuals and their environment, but the individuals themselves do not evolve. So it's only populations that evolve. I put a true-false question about that in the quiz too, and frustratingly, a lot of people got it wrong. I would assume that most of the people who were missing questions like that are the people that aren't in here right now. The second one being that natural selection can only amplify or diminish heritable traits. So any other trait that is not passed on from one generation to the next, natural selection cannot act on it.
Like losing an arm or a leg.
Yeah, like losing a limb in a car wreck, or getting a tattoo or something like that. Not encoded in your genes, natural selection cannot act. And then finally, that evolution is not goal-oriented. That's one of the things that separate artificial selection from natural selection. So there's not some sentient force that is directing evolution of organisms. It's all just a result of happenstance and how things are shaped by the current conditions in their environment. Okay. So that's what I meant by the 3 key points about natural selection. Now, you should also understand why Darwin's finches were so important to his formation of ideas about natural selection and evolution.
It was the beaks, right?
Well, basically what it was is that he'd observed this species of finch on mainland South America, and then he got to the Galapagos Islands, and he saw a bunch of different finches that looked like they were the same finch, slightly modified to serve a slightly different purpose. So they had these different beak sizes, which were well suited for exploiting different sources of food in their environment. So that is why they were so important, because it was such a close relative, and it was so obvious how and why the changes occurred. Does that make sense? Okay. Also understand how those different species came to be. When they were introduced to a new environment, they exhibited, would that be sympatric or allopatric speciation? I guess it also, that depends on your perspective. If you're comparing it to the mainland population, then it's allopatric. But the population of finches within the Galapagos Islands, that's sympatric speciation, because they evolved into different species while occupying the same space and time. What kind of natural selection would have caused that? With our 3 main categories of natural selection, being stabilizing, directional, and disruptive. Disruptive, right? The one that selects against the intermediate phenotype. So let's say when these finches originally came over from the mainland, they all had medium-sized beaks. But then they get to the islands and there's no medium-sized seeds. there's only really big ones and really small ones. The medium-sized beaks are very bad at breaking the large ones, and they're too clumsy to pick up the tiny ones. So then any species, or not even species, any individual that was born with a slightly larger or slightly smaller than normal beak would then have an advantage in exploiting the food sources currently available. So that selects against the intermediate phenotype, the medium-sized beaks, and selects for the really small and really large ones. That's disruptive selection, which is more likely to result in sympatric speciation because you're forcing the two ends of the population farther and farther away from each other until they eventually become reproductively isolated. The big beaked birds are likely to only breed with other big beaked birds, same with the small beaked birds. Make sense?
Would y'all refer to the islands where there are already separate species?
Yes. So this would have happened over a very, very long period of time. You couldn't have observed the actual origination of new species there because it would have taken too long. But remember the example where people monitoring beak sizes do see large shifts in the population over short periods of time. That's one of the reasons we can be so confident that that's actually what caused those different species to arise. Okay, number 7. Everyone should hopefully understand how natural selection leads to pesticide resistance in insects. And it's the same process that leads to antibiotic resistance in bacteria. You should hopefully be able to explain the basics of that process. Is anyone not comfortable with that? Okay, everybody is great. So number 9 then. This is the 3 things that must be true about a trait in order for natural selection to act on it. What are they?
Variable.
Must be variable. There must be at least 2 different forms of the trait. Must be heritable. Must be something that can be passed on from one generation to the next. And finally,
Must contribute to sexual reproduction.
yeah, it must contribute to differential reproductive success, meaning it must make the organism either better or worse at reproducing. So the first 2 things can still be true, and if the third isn't, natural selection is not going to act on it. Like a great example would be, let's say, eye color in people in modern society. If you have brown eyes or if you have green eyes, it's highly unlikely that that's actually going to contribute to differential reproductive success. Neither one of those is going to make you more or less likely to have any kids, right? So that's an example of where a trait can be heritable and can be variable, but still won't be acted on by natural selection because it doesn't make you either better or worse at reproducing. If any one of those things is not true, natural selection cannot act on the trait.
So then how would you describe beak size as being
So if you're better able at getting food, you're better able at surviving long enough to reproduce. All right, is everybody comfortable with number 9? Okay, I'm gonna assume that, I'll assume no answer is a yes. So number 10, you guys remember how to calculate allele frequencies? Does anyone not remember how to calculate allele frequencies? Okay, so it's actually a fairly simple process. It looks intimidating at first, but once you've seen it walk through step by step, it's pretty simple stuff. Do you guys know what Hardy-Weinberg equilibrium is? That is when a population is not currently evolving. So there's no change in allele frequencies from generation to generation. So in order for a population to remain in Hardy-Weinberg equilibrium, 5 things, oops, on screen, 5 things must be true. Where is it? There it is. So the 5 things that have to be true in order for a population to remain in Hardy-Weinberg equilibrium is 1, a very large population. Because if you have a really small population, it's quite possible that some alleles are going to get left out of a generation just by random chance. There must be no gene flow between populations, meaning no individual is either leaving the population or entering from somewhere else. There must be no mutations. There must be random mating.
Before, what would contribute to Parkinson?
So if you have non-random mating, that means that some of the organisms, usually the females, are going to be selecting for certain traits or against certain traits. And if they're selecting for specific traits in picking whichever male they're going to mate with, then that means that males that have certain alleles are going to reproduce more often. That's going to throw the population out of priority 1 breeding equilibrium because that's going to enhance the presence of certain traits or certain alleles in the population. Now, it's almost impossible for a natural population to be in priority 1 breeding equilibrium. It's just something that's good at conceptualizing the factors that cause evolution. Does that make sense? Okay, great. All right. Where are we? Number 12. The 3 basic categories of natural selection and the 2 basic categories of evolution. We just mentioned our 3 basic categories of natural selection. What were they?
Disruptive.
So we have stabilizing, directional, and disruptive selection. You guys should understand what all of these mean and what the consequences are going to look like. So in stabilizing selection, that's when it favors the intermediate phenotype and selects against the extremes at either end. Directional selection is going to shift the population in one direction or the other by acting against one extreme but not the other. And then disruptive selection we just discussed is going to select against the intermediate phenotype, selecting for both extremes, and this one is what's most likely to result in new species. The 2 types of evolution are convergent and divergent. Divergent evolution meaning when 2 species share a common ancestor, but end up becoming different enough that they're eventually 2 different species. Convergent evolution, when species do not share a recent common ancestor, but they occupy the same habitat and therefore develop similar traits. That leads right into the last part of that thing on the study guide, which was homologous and analogous traits. Homologous traits are from organisms that share a recent common ancestor, and they have evolved to serve different purposes, but still share the same basic construction. As in the forelimb of any mammal. Has the same bones in it, but certain bones have been either enlarged or reduced in order to allow that bone to serve different purposes. Analogous structures are the opposite. It's where some structure serves the same purpose, but has a different evolutionary history. So in this case, with the moth, the pterodactyl, the bird, and the bat, they all have wings, but none of them share a common ancestor that has wings. Their wings all developed independently in their evolutionary history, so they have very different basic construction.
I'm still fuzzy on the name. That's like the first plants that we went over today, right? The pines and the redwoods and the sequoias. It's like you said that they
Didn't all evolve from an ancestor of seeds.
Right.
Yeah. So that's the same kind of thing. So the cones of a lot of the gymnosperms would be considered analogous genetics. They serve a similar purpose, but they have different evolutionary lineage. Is everybody cool with those now? Okay. So then moving on to 13. Know what speciation is. That one's pretty obvious. What is speciation?
What are the new species?
Yeah, development of new species. Now also remember the difference between allopatric and sympatric speciation. These are terms used in biology which are basically time and place specific. So if I use the word allopatric, that means things that are occupying a different place and or a different time. If I use the word sympatric, that means they're occupying the same place at the same time. So allopatric speciation is when a population gets separated by usually some geographical feature, like the formation of a mountain range or the formation of a river or lake, and they are no longer interacting with each other because they're not occupying the same space at the same time. That is allopatric speciation. Sympatric.
The finches were, right?
Well, that's if you're comparing the island finches to the mainland finches, that would be allopatric. If you're comparing the arisal of, is arisal a word?
You're right.
Maybe I'm trying to say arisal. If you're trying to compare the development of different species on the islands, then we're talking about sympatric because they're in the same place at the same time. They're just evolving to occupy slightly different niches within that ecosystem. Does that make sense? Great. Okay, and then finally, number 14, be familiar with some of the common misconceptions regarding evolution and know why they are wrong. That was the final slide of that lecture. So one of the common misconceptions is people will try to paint evolution as some uncertain guess by describing it as just a theory. The problem there being that that's using theory in the pejorative or informal sense and not in the formal scientific sense. In science, something that's described as a theory has been vetted by extensive, rigorous research. It's not just something that someone came up with to say, hey, maybe this is it. Another misconception being that individuals can evolve. People miss that on the quiz. People are going to miss that on the exam. Don't let it be you. Individuals cannot evolve. Only populations do. Another being that evolution is supposed to explain the origin of life. So people will try to discredit evolution by saying, well, it doesn't accurately explain where life came from, so obviously it's wrong. It's not supposed to explain where life came from. It's only supposed to explain how life has changed throughout its history since it came from wherever it came from. Also, that organisms evolve on purpose. That's obviously not the case. It's just a result of happenstance. People will also try to paint evolution as controversial among scientists. This is absolutely not the case. There is a broad scientific consensus in support of evolution. And then the most annoying one being that other theories should be taught in schools. That annoys me because there are no other scientific theories. This is the only one.
And the only reason why we call it, or why the science community calls it a theory, is because we can't go back in time and physically see things,
Well,
right?
I mean, gravity is also a theory in that sense. Our best theory to explain why something falls when we drop it is because the more mass something has, the more it curves space-time. That's still a theory. So evolution is a theory in the same sense that gravity is a theory. But I don't hear anyone arguing gravity. Although people are arguing the shape of the Earth now, so who knows, maybe we're just a few years away from that. Anyway, so the last scientific theory that, other than natural selection, that was proposed to explain evolution was Lamarckian evolution. That was the one where the guy thought that acquired traits would be passed on. And we know that is not the case. Although we're also just recently discovering that field of epigenetics, which suggests that things you do during your lifetime can have some effect on the expression of your genes in future generations. Really cool. But that still doesn't mean that Lamarck wasn't wrong. Okay, any questions on chapter 11's study guide? You guys want to go over chapter 12 study guide? A lot of you look like you want to leave. You can go whenever you want. We're officially at the end of class now, in 1 minute. This is now office hours. So, who wants to do chapter 12 study guide? Great. I don't have chapter 13 or 14 study guide written yet, but that doesn't mean you can't talk about it. Now remember, chapter 12 was a relatively short lecture. It's also an extremely short study guide. Right? So one big important thing here is to remember that all life currently existing on Earth evolved from one common ancestor. Head-scratchingly and face-palmingly, people missed that on the is too. Don't let that happen to you. So you should understand that there was that last universal common ancestor, and you should have a very basic idea of when that last universal common ancestor existed.
We would call it no, we would call it cyanobacteria, right?
No. No, cyanobacteria are definitely much, like a very long time after Luca. So it's thought that Luca existed somewhere around 3.9 billion years ago, give or take. I think 3.5 to 3.9 billion years ago is our range of guesses.
Would it be a microorganism?
Absolutely, yeah. It was a prokaryotic unicellular organism. It's also important to remember that we don't think this was the first organism to have ever existed. So life probably originated 4.3 to maybe like 4.5 billion years ago. So there were hundreds of millions of years where other organisms existed, other lineages existed, maybe even longer. So it doesn't mean that just because everything that currently exists evolved from this one organism 3.5 to 3.9 billion years ago, it doesn't mean that there weren't any other lineages existing during that time. It just means that they all went extinct before now. So there were plenty of other organisms existing on Earth at the same time as our last universal common ancestor. It's just that none of their evolutionary descendants have survived until present day. Hopefully you guys know how binomial nomenclature works. That's the genus and species name. Should always be italicized. Genus should always be capitalized. Species should never be capitalized. You should know the basics of taxonomy. You should know all the levels. What's the most general level of taxonomy? There's 3 of them.
Bacteria, archaea,
And eukaryote. And those are domains. That's what we call that taxonomy, is a domain. If we go to the next most specific one, what's that? Within the eukarya, there are 4 of them. Or is it 5? Plant, animal, fungi. Well, actually I take that back because we're just now revising that, so Protista doesn't even count as one. But it's the kingdoms. Kingdom and then 5. So kingdom would be like animals or plants or fungi. Phylum would be something like Chordata, which is our phylum, or Echinodermata, which is the phylum that sea stars and sea slugs belong to. Below kingdom below phylum we have, anybody remember that mnemonic I gave you?
No, I know.
So it was kinky people come over 4 group sets. Kingdom, phylum, starts with C. Class, yes. Kingdom, phylum, class. Then, starts with O. Order. Then, family. Then, genus. Then species. And in some rare cases, there's also subspecies like with dogs. Canis lupus familiaris. But most things aren't classified as far as subspecies. Alright. You should also know the basics of phylogeny. So phylogeny is basically mapping out the proposed evolutionary history of organisms. And if I were to give you a simple phylogenetic tree, you should be able to read that tree. Like, for instance, let me pull one up. Oops, wrong button. So, go to my diversity of life lecture. Alright, so here is the phylogenetic tree of all life that we know of. So you should be able to read this. For instance, if I were to ask you, are bacteria more closely related to, well, I'll say eukaryote. Are the eukaryotes more closely related to the archaea or the bacteria?
Archaea.
Archaea, how do you know?
Because there's the most common ancestors.
Yes. There's
closer
a
to
more
our-
recent common ancestor.
Can you repeat the range over the ground?
Repeat what?
How to answer this question?
Okay, so the way you would answer that question is you would look at how far back you have to go before it shares its most recent common ancestor. So the eukarya, this is their most recent common ancestor with the archaea. You have to go farther back to find their most recent common ancestor with the bacteria. I could also say, are animals more closely related to the halophiles or the flagellates? Yeah, because their most recent common ancestor is much more recent.
But you will give us this map.
Oh yeah. If I were going to ask you that question, I would absolutely have this tree up there. I would not expect you to have this memorized. I wouldn't even necessarily expect you guys to know what the halophiles are in the archaea. But yeah, if I'm asking any questions about a tree, I will give you the tree. Because I don't expect you to memorize these things, I just expect you to remember the basics of how to read. So for instance, let's say I wanted you to name a sister taxa of the animals. You name one? What's a sister taxa?
Plants.
Yeah, plants, or in this case, fungi, because we're not really certain about exactly when they broke off. But, so a sister taxa is something that shares the last common, or the last common ancestor on the tree. So they branch off directly from each other. Also remember though that doesn't mean that one evolved from the other, as with the humans and the chimps. It just means that we evolved from the same common ancestor. You guys fairly comfortable with how to read a tree? Those are the most in-depth questions I would ask you about phylogenetic tree. Alright, so then, number 5. Know what those nodes or branch points are and what they mean. So if we go back to this, here we have a node where things are branching off. What does this represent?
Most common, or most recent common ancestor.
Yeah, there's a few ways to answer this question, one of which would be that this represents the divergence of two different species from whatever this was. You could also say that it's the common ancestor shared by everything that splits off from there, or something like that. But nodes represent significant evolutionary events that lead to different species. Or different evolutionary lineages entirely on a tree like this, which you don't even see any individual species because it's all so general. Just for fun, I'm going to see if I can find a good picture of a more detailed phylogenetic tree of all life. Where is it? Here it is. Alright, I'm going to use this new trick I learned.
So it's just a good chance it'll come out really clear.
Yeah, but it shouldn't, because this is a high-res image. So what's going on here? Yeah, you're probably right. So, well, let's just leave it up like this. That won't let me enlarge it.
Sometimes.
This isn't all that important anyway, but there you go. This still doesn't go down to the species level. I think as far as this goes, I can't even read that because the print is too small. But
That
that's,
doesn't really show most common recent ancestry, does it?
it does. So on this thing, looks like right about here would be Luca. And then everything branches off from there. The cool thing about this tree is it also gives you a time scale. Remember that most phylogenetic trees don't do that. The events are in the right order, but scale is not meant to represent time. So like, if you're looking at this phylogenetic tree, if I were to say how much time passed between when the archaea branched off from the bacteria and when the eukaryote branched off from the archaea, you would have no way to answer that question because this isn't scale over time. You would just know that it happened later. For the most part, we don't try to include time scale on phylogenetic trees because it's very difficult to do.
So most bacteria has already gone through most of their evolution?
I'm not sure what you mean.
Like, because the time frame of that bacteria is all towards like the earlier portion of the time scale. And so are they not evolving anymore?
Oh, they're definitely still evolving.
Yeah.
So that's not to, like, also bear in mind that when you have something branch off, that doesn't mean that other things have stopped evolving. So on this tree specifically, if we're talking about the plants, so the plants branched off here, that doesn't mean that this lineage is no longer evolving. Yes?
Go ahead.
Go ahead. No one is required to be here.
Do it because we gotta go
All right, good luck. Yeah, lab starts at like 2:30, doesn't it? 2:25. Okay, you got 4 minutes.
I'm on Tuesday, so.
Good. Okay, so then I guess we're on to number 6, remembering the basics of how phylogenetic trees are constructed. So do you remember what types of information are used for that?
Not specifically.
So basically we use everything at our disposal for this. It's become more accurate with the advancement of molecular biology. So we can use amino acid sequencing, we can use gene sequencing, stuff like that. But we also still do employ morphological characteristics as well.
Okay.
Like, for instance, that's how we know that humans and fruit flies aren't necessarily super closely related, even though we share about 60% of our DNA sequences. So in the past, it was done entirely morphologically, or almost entirely morphologically, which is why we have a lot of errors in there. But the new molecular techniques have allowed us to really refine. Okay, and finally, know how to determine whether or not two species are closely related. So I'll go to the primates tree here. So this one's fairly simple. If I were to ask you whether the humans are more closely related to the bonobo or to the Bornean orangutan, should be relatively easy to figure that out because we have a much more recent common ancestor with the bonobos. So that's generally how you would tell whether or not things are closely related, is by looking at how recent their most recent common ancestor is. Okay, so that's the 2 study guides I have up there.
And
What
then,
else?
chapter 13 was evolution, right?
Which one?
Chapter 13 was evolution.
Chapter 11
No.
was evolution.
Right.
Chapter 13 was diversity of microbes, protists, and fungi. You have any questions about that chapter?
No. I mean, I did pretty good on the quiz.
Yeah, you did.
Yeah.
I will try to have the study guides for 13 and 14 up by tomorrow at the latest. Hopefully.
And the most interesting thing was finding out that fungi are closely related to us.
Yeah.
We never thought that.
Nobody ever does. I didn't either until I learned it. I was pretty blown away that fungi are more closely related to animals than plants are.
I have a few buddies that are really into fungus, and they were telling me things like that, but I never knew why they would say that we're like close relatives and stuff.
Yeah.
Like,
Yeah, fungi are also more closely related to us than they are to plants, which is pretty cool stuff.
cool. Thank you so much.
No problem. We'll see you on Tuesday.